Good evening, and thank you for joining us for our 2025 White Mountain Research Center Lecture Series, celebrating 75 years of scientific achievement. My name is Kayleen Kinsey, and I am the scheduling coordinator at WMRC, and I'll be the moderator of this lecture this evening. Some housekeeping items to remember is to please silence or turn off your cell phone, and we will have a Q&A after the talk this evening. With that being said, well, before that, this talk is being recorded on several platforms, and it will be posted to our YouTube channel at a later date, which you can find on our website. And with that being said, I would like to begin by giving a land acknowledgement. The White Mountain Research Center acknowledges the Numu, Paiute, and Niue Shoshone as the first peoples and traditional land stewards of Paiute and Kohotoya, the Owens Valley White Mountains. As a facility managed by a land-grant institution, we pay our respects to the elders, our relatives, relations, and friends of past, present, and emerging generations. Excuse me, I'm getting over a cold. I would like to introduce this evening's speaker. Nathan Rank is a professor. Dr. Nathan Rank is a professor of biology at Sonoma State University and a longtime researcher associated with White Mountain Research Center, who once began here as a student researcher himself, working under the former assistant director, John Smiley. The title of his presentation this evening will be Insect Population Ecology in the Sierra Nevada, a 40-year perspective, where he will be discussing how willow leaf beetles tolerate and survive exposure to environmental extremes in the Sierra Nevada mountains. This is the longest running study at WMRC, which began in 1984. And with that being said, Dr. Nathan Rank.
All right, thank you, Gayleen.
You're welcome.
Before I start, I'd like to thank John for introducing me to the system and for being a wonderful advisor for years, not just during grad school, but later on. And I'd also like to thank the research station for supporting this work for so many years. My first year, I got $300 to spend 6 weeks here, and I made it work. And I loved it here. So I didn't think at the time that I'd be here so much later. And as I was thinking today, when's the last time I gave a talk at White Mountain? It was like, oh, that was 37 years ago. And then no, wait a minute, there was another one 22 years ago. So it's been a little while. It's great to have a chance to talk to you. I'll warn you that this talk is exceedingly fresh. It's just got out of the pizza oven. And so not sure how long I'll take. And I'm not sure at what level it is, but hopefully it will be reasonable. I did have someone check it out a little bit. Okay, so we're going to talk about ecological and evolutionary forces promoting biological diversity, which I decided is kind of a motivating force in my work and has always been, so I sort of tied everything together around that framework. I'll talk a little bit about my PhD work on the ecology of host plant use in leaf beetles, which is part of a research project that had to do with what forces might maintain and enhance biological diversity. Then I'll talk about the role of microbes in reproduction and geographic variation. And so when we talk about ecology, we're talking about the ecology of multicellular organisms, insects, and other animals that we're used to, but also about the single-celled organisms that are interacting inside of the insect that we study. And then I'll talk a little bit about the California Conservation Genomics Project, which is a statewide project designed to document the biological diversity of California using model species and using that information to help inform management plans and stewardship. And so it's a great project, and you'll learn more about it in a bit. Okay, so we'll start with a quote by Charles Darwin. There is grandeur in this view of life with its several powers, having been originally breathed into a few forms or into one, and that whilst this planet has gone on cycling according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. So this was the concluding sentence in The Origin of Species. It's been used as book chapters and book titles, so you've seen some of the phrase of this before, but it again kind of has informed my work even before I started college. I was interested in, of course, I was interested in dinosaurs and those kinds of things, and evolution, and so let's talk about factors that promote biological diversity. First, we'll talk a little bit about the ecological scale, so the scale of interacting species in the present day. What are some features of the abiotic environment that are associated with more and less diversity? What is the role of environmental variation in space and time? Then we'll talk about the evolutionary scale of processes generating biological diversity. For example, time for formation of new species, role of environmental variability again, and interactions in coevolution with other species. And this word coevolution is the word, sort of the driving word that this talk revolves around, because I got interested in coevolution before I started grad school, and I found myself studying it in a very different way than I originally expected to. So let's talk about the abiotic environment first. Diversity tends to be greatest in environments that are intermediate. If environments are too physically stressful, then you create first physiological stress for organisms living in those environments, and at some point you get beyond the capacity of organisms to survive. So the optimal conditions for different organisms will vary, but within an environment you tend to find lower diversity in places that diverge more from the optimal conditions for the species living there. One way you see this play out is in latitudinal gradients, and I think this slide was latitudinal gradients of amphibians, so the patterns vary a little bit depending on the taxon you're looking at, but we tend to see higher diversity in more stable environments near the equator, and reduced diversity as we go out towards the poles, where abiotic conditions can reach the point of physiological stress or even inability of organisms to live there. Found a nice altitudinal gradient using Mont Blanc, and so you can interpret a little bit of the French here, but we also see similar gradients in diversity and abiotic conditions in montane environments, as we know, on either side of us here, and when we get to certain elevations, we get to the point where plants and hardly any insects can make it, and of course the elevational ranges in the Alps are about 1,000 meters lower than what we have here, but in any case altitudinal gradients can often replicate latitudinal gradients, and specifically the organisms I study they're found a lot more in higher abundance in the far north than they are found in California. And so here in California, they're living in the relict regions where conditions are somewhat similar to what they experienced further north. So there's also a component of the middle is best with respect to levels of environmental disturbance. So how frequently conditions are reshuffled or some major change occurs, for example, fires or floods. Grazing is another example. So there's a gradient of disturbance, and the overall consensus among ecologists is that diversity is greatest at that intermediate disturbance level, and that at lower levels or at high levels of disturbance, you lose species because conditions change and they can't survive and thrive and reproduce. And so abiotic factors are more important on the left side of the graph, and then on the right side of the graph, if disturbance is too infrequent, you tend to find a few competitively dominant species predominating and excluding others, so lower diversity. Okay, so we've got the ecological scale. Now we're thinking about the evolutionary scale. And for this, it's worth reminding us that Earth's biological diversity over the last 500 million years has been punctuated by frequent mass extinction events. And those mass extinction events are associated with substantial reductions in many cases in the diversity of organisms belonging to major groups. And they also show that it takes some time to recover from those mass extinction events. So what's on the y-axis here is extinction frequency. If you sort of have to flip the graph on its head to think of it as the effect on biological diversity. And you can see one of the biggest mass extinction events, even bigger than the KT boundary, Cretaceous-Tertiary boundary, where we know a meteorite was involved, was at the end of the Permian period. And Owen could probably tell you more than I can about why that was such a severe mass extinction event, but that'll have to wait till after I've finished. So the effect of the Permian extinction on insects was very substantial. So now we're talking about insects themselves, and we're used to thinking of, you know, the beetles that, or I mean the insects that have complete metamorphosis as being the most diverse insects on the planet. Well, that might not have been true if it hadn't been for that Permian mass extinction, where other insects that have externally developing wings and no complete metamorphosis were of greater diversity than the holometabolous insects that we see predominating today. So mass extinctions cause a reshuffling, and there's some duration in between the extinction event and the accumulation of diversity. Okay, two groups of organisms that this laser pointer off. Okay, so there have been two ways that species interactions have been considered to be especially important for the generation and maintenance of biological diversity. And these two mechanisms both concern coevolution. So changes, genetic changes in one species that drive changes in another species, which then may reflect back and cause change in the first species. And so these two hypotheses, one is called the escape and radiate coevolution hypothesis, and it was proposed by Ehrlich and Raven to explain the high current diversity we see of flowering plants and the high diversity of insects that we see on the globe. And so this idea was developed by looking at butterflies and their patterns of host plant use and age. And the notion was that the two groups have caused each other to form new species by escalating new kinds of defenses and counter-adaptations against those defenses. So that's the first kind of coevolution. The second one is that microbes can be so closely associated with their hosts that they actually affect the reproductive biology of their hosts, and they can generate incompatibilities, which then turn into reproductive barriers, which over time could lead to generation of new species and therefore enhancement of biological diversity. So here we are with our flowering plants. I'll talk about escape and radiate first. Diversity of insects. So two of the biggest groups, terrestrial living organisms we have on the planet today. So in the geologic record, we see that pattern that I was alluding to. Let's see here, this graph get a little confusing here. But the main point is here on the left, that flowering plants began to diversify in the Mesozoic during the age of the dinosaurs, and then after the Mesozoic extinction, they radiated quite rapidly, diversified, and insects have sort of followed along, and again, you saw some of the patterns earlier, but especially it seems like insect diversity has risen in concordance with the diversity of plants. And then this figure just shows how that breaks down into individual groups of insects belonging to those main orders that I mentioned earlier. And then we have the VGA cable. Okay. So this Ehrlich and Raven hypothesis is you can sort of draw out the scenario using evolutionary trees. So these are phylogenies. Nowadays, phylogenies generally are made using patterns of DNA sequence changes, but you can use fossils or morphological traits to generate them as well. And so the idea here was on the left, you see a lineage of plants. So this represents 1, 2, 3, 4, 5 lineages of plants, and these plant species on the lower part of the figure are being consumed by certain kinds of herbivorous insects, maybe specializing on that group. And the notion is that when the plant evolves some kind of innovation, some kind of new defense trait, which is often secondary chemicals that can be toxic to herbivores, that provides them with a new adaptive zone that they can then speciate into, and that therefore the evolution of this trait is associated with greater diversity in the plant. And then along the same lines now, eventually the insects will evolve some kind of counter defense, some way of overcoming that defense, and they too begin to diversify onto these new, more chemically defended plants. So this is the Ehrlich and Raven model. And when I first started coming to work on the willow leaf beetle, I was thinking about that model. And I actually was supposed to work on tropical butterflies, and had a little trouble getting into the OTS course to work in the tropics. And John said, well, you know, the White Mountain Research Station offers some mini grants to grad students for the summer. So that's how I got the $300. That's why I'm here today. Okay. So, but this genus, Chrysomela, is actually a genus well suited to trying to understand the evolutionary processes behind diversification of a group of insects. When you look at these species of beetles, you'll see that our species here in the Sierra has some pretty similar looking close relatives distributed across the globe. And so most of them are concentrated in North America, but we do have Chrysomela lapponica in Europe. And what you can see is that these species, despite their very similar morphology, have different host plant use patterns. So they feed on different plants, and those of you who know, you can chew on a willow leaf and deal with a headache. Nobody's going to chew on a birch or an alder leaf in order to deal with a headache, right? They have substantially different leaf chemistries and are much more distasteful. But these beetles seem to be switching back and forth between them. And one of the things that the guy who did the monograph on these in the 50s did was he actually crossed them, and he found that he could obtain offspring that could feed on both of these chemically very distinct plants. So this is a great candidate to study the evolution of host plant use, partly because, as you'll see, we can study the ecology of it, but also because the group itself seems to be involved in some kind of diversification of host plant use process. Okay, so we have covered the first item. Now we're going to talk about my PhD work. And we'll start with work by some others, including Jacques Pastilles in Brussels, and Elizabeth in the back knows Jacques as well, so this is a figure from one of his papers. He worked this system out. So our willow and poplar feeding Chrysomela larvae extract salicylates So the genus is Salicaceae. So salicylates are chemicals that are characteristic of, did I say genus? Family. And Salix is the genus. So these chemicals are characteristic of this plant family. Salicin has a glucose, and this organic ring compound here, terminology is failing me right now. In any case, the larvae of the beetles feed on the leaves containing the salicin, and they, of course, absorb food into their hemolymph, and waste products are excreted. But the salicin converted to salicylaldehyde is then sent to a reservoir on the dorsal side of the beetle, and when you disturb these beetles, they release these droplets of defensive secretion, which are very irritating smelling, and you could easily imagine that you would not want to eat one of those larvae. It smells like a nasty organic compound. Well, so the willows here in the Sierra Nevada, and in the White Mountains for that matter, turn out to vary greatly in the concentrations of those compounds. I had to extract this table from my PhD thesis, which I managed to find online today. The Sierra willow has 150 milligrams per gram of leaf tissue of these salicylates in them. The next species, Salix lutea, which superficially looks pretty similar, it takes me a while to explain that to students. Well, I make them eat the leaves. That's what I do. But in any case, very low levels are absent of salicylates. And then the Geyer's willow, Salix geyeriana, is sort of intermediate. And then we have a couple more willows. Of course, we have more than that. But in any case, you can see that the plants vary greatly in their chemistry, and they grow together in these high elevation bogs. And so this looked like a great environment or habitat to investigate the effect of the variation in host plant chemistry, not only on the beetle, but on the natural enemies of the beetle. So, the prediction is that the plant chemistry, which, by the way, you can see the herbivore overcame that plant chemistry to develop this adaptation. So the plant chemistry generates enemy-free space, and the idea would be that if the beetle larvae are feeding on salicylglucoside-rich willows, salicylglucoside is the fancy name for the salicylates, then you would predict that they should prefer those plants because that's where they get their secretion. You'd predict that predators should be repelled by that secretion, and beetles should survive best on plants with compounds they need for their secretion. And so to test this prediction, I did some lab experiments over there in the Manus lab, and I also did some studies of beetle abundance in the field. What I found was that Salix arrestra, the one that is the highest in salicylates, is also the preferred plant in two-way tests. I could get them to eat pieces of filter paper with salicin in them. I added a little sugar. So they're stimulated to feed by salicin. And in the field, their abundance tends to be greatest on plants with higher levels of salicylates. So first prediction came out pretty nice. I hit a wall on the enemy-free space hypothesis, though, when I started studying the nature of their predators. And so it turns out that the predators of these beetles are actually specialists. They don't eat anything but, in the Sierra Nevada here, Chrysomela aeneicollis. So the first predator is Parasurface melanderi. These are beetle eggs, and the little white egg shells between them are the eggs of a maggot, the fly, Parasurface melanderi. So she lays her eggs in between the beetle eggs, feeds on the eggs, and then starts roaming around the plant and consuming any larvae she runs into. This is actually a video I took at the station here, and you can see the beetle putting out its defensive secretion and the maggot just going and going and going, getting its mouth hooks on the underside of the beetle larva, paralyzing it, sucking it dry within 15 minutes. So this turns out we did some studies showing that they're attracted to the secretion. In fact, Jerry, was involved in many days of testing pieces of filter paper with beetle secretion and examining the behavior of the syrphids. The second species, Simorphus cristatus, is also a specialist. This is a picture John took, and it shows you a wasp stuffing a beetle larva into her nest. And so turns out they only eat third instar beetle larvae. Then we've got a couple of parasitoids. Parasitoids are insects that lay their eggs on or near an insect host and then burrow inside and eat the insect from inside. So those tend to be specialists as well. So we got 4 specialists, and none of them seem to be repelled by that secretion. Some of them seem to be attracted by it. And then, so the first summer that I actually did a field experiment, I put larvae on leaves near the cabin in Big Pine Creek, and tracked their survival over the course of the summer as they developed from young first instar to middle-sized larvae and then reach pupation, which took about 40 days and ends in August. So this is a lesson. If you do a field experiment, if you do it once and you want to run away with your hypothesis and say, yes, I proved it, don't do it again. And then don't do it again. And this doesn't show the 4th year where I expanded the design and added 3 more species, and it still showed no effect. So of the 4 times that I ran the experiment, I only got a difference once. I really like to punish myself, so I went to Finland a couple years later and did it again with a different beetle and got the same answer. So not much. Okay, well, here we go. So beetles do prefer SG-rich plants, stimulated to feed by them. Natural predators don't seem to be repelled by the secretion. Some are maybe attracted to it. Survival is not greatest on those plants. So beetle host plant use may evolve due to other factors besides natural enemy pressure, and plant chemistry alone may not be responsible for beetle diversification. Now, one of the things that I also discovered, because I went to Europe for my postdoc after getting to know these species here and working with them, and I started working with similar species in Europe, related species, and I did some library work, and I found out that of the predators that we see here in the Sierra Nevada, so that's the middle here, they're also found in Eurasia. So in the case of the Parasurface, people had not really paid much attention to it. It was in the older literature, 80, 100 years old, but people didn't realize how important this was as a predator on related beetles to the ones I study here. And so everywhere I go, I'd find a egg clutch of beetles and I'd find a fly on them, boom, new record. So they don't specialize globally, but they do specialize, especially here in the Sierra where they only have one prey item. Same thing with the Simorphus wasps, and they also have tachinid flies and related parasitoids and other things. The main thing that you see in Eurasia that you don't see here is a variety of predatory bugs and ladybird beetles that are specialized on these kinds of leaf beetles. And I think that what happens is our abiotic conditions here are just too harsh to support a robust predator community. So we have a few of the major ones that are most successful. So that, I guess the lesson of that is the entire community, the entire food web, the biodiversity of it here is exceptional, but it's part of a global community. And so what we see here often are things that are closely related to things that you might see in Northern Europe. It's true for the willows too. Okay, so we're going to turn to microbes. I have no idea how we're doing time-wise. Good? 30 minutes. Okay. All right. So we're turning our attention to microbes. So let's look at this for a minute. Some species of bacteria are passed from mother to offspring, especially in arthropods, including insects. So these are not bacteria that you normally catch or pass on to another individual in the same generation. They are inherited along with the nuclear genome and the mitochondrial genome. Turns out they pass only down through the female line, and that generates some selective pressure on those bacteria to co-opt or modify the reproductive systems of their hosts. And so these bacteria, especially Wolbachia bacteria, can generate reproductive barriers in hosts that cause them to form genetically distinct lineages that eventually become species. I'll have a bunch of slides to sort of demonstrate how that happens in a bit. So it also turns out that these beetles here, and I'll give you details in a little bit, host multiple lineages of these kinds of bacteria that are passed down from mother to offspring. And so Wolbachia, again, is known from arthropods and nematodes. It is an intracellular parasite, so it lives actually inside the host cells. As we'll see in a minute, often you have multiple strains in beetles, and they do some wacky things. In some cases, like ladybugs in the UK, the sex ratio is 70% female in a natural population because the male embryos were killed by the bacteria. Now, why would the bacteria kill all the guys? Well, for the bacteria, those guys are dead ends. If she passes them on to a male, it's good for the beetle to have enough males around, but for the bacteria, the male is a waste. And he'd rather have all that energy, or the bacteria would rather have all that energy invested in more females, which can then transmit the bacteria to more offspring. So that's one way they can do it. They also sometimes, in the case of not insects, but in isopods, they will turn the isopod from male into female. So they feminize the host. And in other cases, they just get rid of males entirely. So you may know that there's some insects that produce offspring without males, and Wolbachia tends to play a role in that. Okay, so the other lineage of bacteria that we're not going to talk about quite as much is Spiroplasma. They're really different in Wolbachia in a lot of ways. Spiroplasma can be found on plants. You can read about Spiroplasma infections of humans. They live outside of the cells in hemolymph, but they also have, in some insects, this property of being transmitted from mother to offspring and killing male embryos. And it turns out they're also interesting because they tend to provide some resistance against other parasites. So a little more about Wolbachia. These are the main lineages of the Wolbachia bacteria, and the figure on the left is labeled mutualist and reproductive parasite. And so what you can see is that in some nematodes, mini nematodes, the Wolbachia is considered a mutualist. And in fact, in some cases, they need the Wolbachia to survive. In cases of insects, they're considered reproductive parasites in the way that I just described, do things that are in their interest and not in the interest of the host. But you can also see a little bit of green here saying that sometimes in insects they also can act as mutualists. So these are bacteria that co-evolve tightly enough with their hosts that in some cases they provide metabolic services to the host. So I wanted to also just mention briefly, you can read about the importance of Wolbachia in biocontrol, not only, as I'll talk about in a minute, in mosquitoes, but also in nematodes. They are actually, you can treat certain diseases in Africa, and I think river blindness is one of them, by administering antibiotics which kill the Wolbachia, not the eukaryote that the river blindness is in, not the nematode. So killing the Wolbachia is good enough. Okay, you can see here, this bacterium has spread so widely in insects that in the family Chrysomelidae, which our target organism belongs to, most or only half of taxa have only one strain. More than a quarter of them have two strains, and a little less than a quarter have up to more than two strains. So they are inhabited by 3 different strains of these Wolbachia bacteria. Crazy, huh? So cytoplasmic incompatibility is one of the ways that Wolbachia and Spiroplasma sometimes can affect the reproductive biology of the hosts. So what is cytoplasmic incompatibility? We got a pile of bugs over here, but we see these little human-shaped icons. So ignore the fact that they're human-shaped, turn them into something else. So a female that is not infected can mate with a male that is not infected and produce offspring. A female that is infected can mate with a male that is infected and they produce offspring. A female that is infected and a male that is not infected, well, all of her offspring get the bacterium from her. So that's fine. But if the male is infected and the female is not infected, all the offspring die. So this is the process that helps facilitate the spread of Wolbachia in a population rapidly. So those individuals that aren't housing the bacteria go down in frequency in the population pretty fast. And so this slide shows you an example of biocontrol of dengue fever in Australia. They have found this as an alternative, treating mosquitoes with Wolbachia as an alternative to genetically modifying those mosquitoes. So add Wolbachia to the population. You could be in a population with hardly any Wolbachia, and within a few months, all of the mosquitoes have Wolbachia. So it spreads that rapidly, partly through this cytoplasmic incompatibility. So males without it mate with females, no kids. So that pushes up the frequency rapidly. Okay, and turns out that those mosquitoes infected with Wolbachia can't host the pathogen that causes dengue fever so well, so it actually reduces the transmission of dengue fever in human populations just by adding this bacterium. Okay. So how could this generate reproductive isolation? How could this help form two new species? Well, if we had a lineage that has no Wolbachia, one strain is introduced to some individuals in a population, and we have another population where another strain is introduced. And then what happens is the pathogen spreads rapidly in each of those populations. And if there's cytoplasmic incompatibility, it's not good enough that they're infected by a bacterium. They would have to be infected by a genetically similar bacterium. And if the two strains are different enough, they will not be able to produce kids. And that generates the potential for reproductive isolation and the formation of new species. So that's what we're seeing on this figure here. And again, the original species just has the original strain, add a new strain, and you could generate a distinct population. Okay, so why is this relevant to us? Well, it turns out that these beetles here in the Sierra, Michael Torelli, who worked out the dengue fever thing and did all the fancy math, said to me after he gave a talk 10 years ago, "Nathan, you must study Wolbachia." And so I said, okay, I better get started. And so we started by just doing gene amplification studies where we add primers of the bacterium to the beetle DNA extract. And the primers are either specific to Wolbachia A or Wolbachia B. Looks like something got a little fuzzy there. In any case, you see bands for both. Same beetle, two strains. So we now know that our beetle has two strains. Then when we look more closely by doing a DNA sequencing study, we find out that within one of the strains, there's actually a second strain. So these peaks show us the nucleotide as it rolls out of a sequencer, and it reads out the nucleotides in a strand of DNA. And if you see two peaks, that means there's two different genotypes there. These bacteria are haploid. If you see two peaks, that means there's two strains. So I had a student work on doing PCR and sequencing from beetles from Rock Creek and Big Pine Creek, and found that there were some differences between the drainages and the amount or the diversity of the strains. But what this shows you is the number of beetles tested, and in this case, the number of beetles with two strains, one Wolbachia A and one Wolbachia B. So they only had two, so to speak. Then the number of beetles with 2 strains of Wolbachia and 1 strain of Wolbachia, so a total of 3. There were 5 Rock Creek beetles with that and 3 Big Pine Creek beetles. Now we get down here, and in Big Pine Creek, you had some with only 1 Wolbachia and 2 Wolbachia B, and then you had some with 2 Wolbachia and 2 Wolbachia B, so 2, 3, or 4. Nobody has none. Everybody has something, and almost everybody has 2. So we decided, well, this was during a period of population decline. Elizabeth will tell you a lot more about that on Thursday. And so we were so desperate to find beetles from a southern population that we went to South Tuttle Creek. Jackie Leary told us about this population, and thank God she went there with us, because that's the worst trail I can think of. So she was looking for bighorn sheep, by the way, and she found one, and while we were there lying there half dead, she was running up the hill looking for the sheep. So we collected these beetles and did some mating experiments, which again Elizabeth may tell you something about, but we also got their genomes, and in the genome files you can find the microbes. And so what this is a lot of work to estimate the density of the 2 kinds of Wolbachia, the 2 main lineages, and Spiroplasma. And you can see that Spiroplasma is present in Rock Creek and barely present in Tuttle Creek. And in contrast, Wolbachia B and A are more common in Tuttle Creek than in Rock. So there's a north-south gradient in the abundance of these microbes. And then if you look more closely and characterize the genetic diversity of the microbes, well, it turns out there's only every Wolbachia B, every beetle has the same Wolbachia B, so that one's pretty genetically uniform, at least in this sample. But within the Wolbachia A strain, the Tuttle Creek beetles, there were 11 strains, so what this shows you is a beetle with a strain. So this strain is different from this strain, is different from this strain, and so on. And so some of the there was one Tuttle Creek, or there was 1 strain in 2 Tuttle Creek beetles and no Rock Creek beetles. And then there are a few strains shared between the 2. And so what this again shows us is that the Wolbachia is genetically diverse. It's not all 1 strain, especially Wolbachia A. So we have a little bit more stuff that we've done with this, and we have evidence for reproductive incompatibility. So we did some antibiotic treatments and found that when we treated one sex and not the other, no more kids. They were laying eggs happily, and we added the antibiotics and they stopped laying eggs, but not when they belonged to the other treatment where they both got the antibiotic treatment. So killing them in both sexes restored reproductive function. We also found that it looks as though maybe one of the Wolbachia strains is a parasite that causes egg incubation to slow down. You don't really want your eggs exposed to that syrphid longer than you have to, right? And so having a longer hatch time is potentially a problem. But ironically, the 2 Wolbachias went in opposite directions. So it looked like maybe one is acting more like a mutualist and the other more like a parasite. And so maybe these bacteria might be involved in creating genetic diversity in their host by generating these reproductive incompatibilities. And so now we're going to talk about the California Conservation Genomics Project and geographic variation. Okay, why care about local adaptation? So we're investigating the potential for local adaptation, and the first step is to scan the genome to look for signatures of genetic differentiation, and then the next step is to look for genes that are especially highly different, which indicates that natural selection, a different evolutionary force, is acting on those genes. Okay, so we want to know something about genetic mechanisms behind the adaptation to environmental stress. Again, you'll hear a lot about that from Elizabeth. We could also use this information to help predict which populations are most at risk. And we could use information about local adaptation and genetic diversity to inform management plans. Now, I don't know about you, but I've always wondered, you know, what was it like 10,000 years ago when those glaciers reached the Owens Valley and there's a big lake here? Where were the beetles back then? Well, they probably were down here, And so as the glaciers retreated, the beetles retreated up to higher elevations where their habitats persist. And what this slide shows you is a couple of genes that we have shown that show a very high degree of divergence between Big Pine Creek and Rock Creek. So two drainages that, as you know, are not that far apart. And the middle drainage, Bishop Creek, seems to be a mix. So maybe beetles are coming from the south and from the north into that drainage, because Rock Creek beetles, again, are very distinct. Now, the genes at which they're most distinct are shown on the map. This shows you the rate of genetic divergence over distance in, it says square root meters. I think that should be kilometers. So 10 anonymous loci show a much shallower rate of divergence among populations than PGI and CO2. CO2 is a mitochondrial gene, and PGI is a gene essential for metabolism. Again, you'll hear a lot more about this on Thursday, but PGI, as soon as we first identified it as an unusual gene, we did a large body of work comparing genotypes at that locus with respect to physiological processes, and we found some very interesting patterns. Basically, the upshot is that the northern beetles seem to be less thermal tolerant, less tolerant of thermal extremes. The northern drainage is colder based on our 20-something years of data logger data. And the southern drainage has a greater thermal tolerance, and so the PGI alleles seem to be adapted to local thermal conditions. The CO2 gene is on the mitochondrion, and so it's tightly linked to other genes on the mitochondrion. So you might expect to see other regions of the mitochondrion showing high differentiation if CO2 is showing that. Turns out there's some very interesting dynamics between mitochondria and the nuclear genome. Even though we've housed them for 2 billion years, they're still evolving and creating problems. Right? Look up mitochondrial disease in humans and you'll read about all kinds of scary things. And so our coordination of our metabolism through mitochondria and our nuclear genes is an ongoing process. And that happens because the mitochondrion is mutating quickly and the nuclear genome has to catch up. So this shows you some mitochondrial genomes from Big Pine Creek and Rock Creek and Bishop Creek in the middle. And I don't want to get too much in depth here, but it turns out that the difference between north and south is in some regions that are fundamental for metabolism, but not genes that code for proteins. So this is not a story like the malaria story where heterozygotes have a superior resistance to disease and things like this. This is a story of how small genomic regions that code for small gene products that are involved in protein synthesis seem to vary among the populations, and that could create some interesting physiological differences, which again you'll learn more from Elizabeth. So those are the two loci that we spent many years studying. When a few years ago, Brad Schaefer, who was my second advisor after John, so I had two graduate advisors at Davis, Brad was my main advisor. Brad has organized this project, which is kind of rolling along, getting a little close to completion, called the California Conservation Genomics Project. And you should read about this project on the webpage sometime. It has a large number of people from a lot of different universities, and Brad should be in that picture somewhere. There's Brad. And so I'm going to tell you a little bit about this project. The idea is to sequence individuals from species across the state of California to get their whole genomes. You can see that the plan is 250 species, plants, animals, marine organisms, marine algae. I don't know about fungi. Are fungi in here? In any case, the individual genomes of the individual species are collected and genome sequenced in a consistent manner. So the idea is to get species that are distributed across the state to mine their genomes at about the same level of resolution, and then collate the data together, and with the overlay of the genetic variability of the different species, you can start to identify hotspots of biodiversity in California. And you can use that to help policymakers make recommendations about management. And so Chrysomela aeneicollis got into the study. Partly because White Mountain has helped us to come back every summer for the last 30 years. And so turned out that there are the Sierra Nevada populations. One population that Elizabeth's ex-husband, late husband, found at Point Arena. And so he identified a population that was totally unknown before he found those. So those are in the sample. Some other populations from Northern California are in the sample. And when all's said and done, an undergrad at Berkeley did this amazing job at synthesizing all the information from this study. And this shows you the ways that the populations in different parts of the state are distinct. So this is the Modoc population, this is the coastal population, really different from everyone else. Within the Sierra Nevada, and there in the whites, that's another place I'll never go back to, 2 days to get there. So a lot of genetic variability. Yeah, right. And when you use that to define genetic clusters, shows us that Tuttle Creek, remember that awful place down, beautiful place down in the south, and where we got the microbes for the beetles, has its own distinct lineage of Chrysomela aeneicollis based on nuclear genes. Then we get this transition through Taboose into Big Pine, which has a different group. Baker Creek, those things we only got there once or twice and they went extinct, so that's an old sample, but you can see that they're an intermediate between the Rock Creek lineage and the Big Pine lineage. It's Bishop Creek that is of mixed ancestry. And then, of course, we get distinct lineages here. I've shown this to people who study species boundaries and how to define them, and they're like, yeah, this could be multiple species. So we may have multiple species of Chrysomela aeneicollis that have been detected through this genome work. And this just shows us that the coastal populations are really different than the montane ones. We think that's a consequence of adaptation to the mild winters of coastal California in contrast to the ones that we have here. Okay, so I didn't know how much further I had to go. Diversity of insects in Sierra Nevada reflects evolutionary history and range expansion. Mitochondrial nuclear genes show evidence of dispersal and hotspots of genetic diversity. The California population support a diverse microbiome that may promote species formation and greater biodiversity. And so the idea is that the microbes that I told you about might be generating these patterns of high differentiation among populations that are essentially in pretty similar habitats. And a lot of people have been involved in this work, and I made sure to thank John right away because I don't see his picture here. So there we are. Thank you. We have, by the way, some of the predators in a box over there in the insect drawer. I could show you if you want to see them. And again, I have no idea what time it is or how long it took me. So.
5 minutes.
Oh, good. Okay, Owen.
Have you looked for the species in other parts of Lake Salinas? So you have Point Reyes that was ripped off of the Sierra Nevada batholith, right? That used to live down in the Mojave Desert and has been since by San Andreas translated up to its position And you only have them at Point Reyes or any of the other parts?
Point Arena.
Point Arena, sorry.
Yeah.
Point Arena. So again, part of.
Oh, so Point Arena is part, oh, no, I hadn't even thought of that, yeah.
This all used to be down in the Mojave Desert, right? And then 30 million years ago, it got ripped off and shoved up north. So I was just curious if in Santa Cruz or in Point Reyes or any other place, you only there, only in Point Arena.
Yeah, so there's that is the southernmost extent. Before we found them there, on the coast, the southernmost extent was Cannon Beach, Oregon.
Okay.
So that's like a 1,000-mile jump there, and I did go down the coast looking for them in Oregon, and we have looked for them extensively on the California coast. We don't see them there.
You don't think they like got there some other way? My only thought was just, have they been there this entire time since being part of the Sierra Nevada a long time ago, being rafted up 30 million years ago?
Probably not. They look more closely related, and some genetic data say they're more closely related to the Oregon beetles than anything else.
It looks like they have no relation to the ones that you have here, but how long does that mean?
Well, they are related. I mean, they are the same, well, maybe, same species. If we put them together, they will mate.
We couldn't,
Okay.
Yeah.
we couldn't get the CCGP to sequence the Cannon Beach population
Okay.
because they were Oregon. We probably could have gotten them to do it if we just like coded them wrong.
Yeah.
But, my guess is if we had those populations in there, they would
That they would be
probably
closer.
group with that.
Okay, interesting.
Yeah.
That's cool
yeah.
to know that geology. I had no clue.
Yeah, that is cool. And you know, I did have a student do a few genes. We had another study. So basically, we did a study that got some from Colorado and Montana. Their real center of abundance is Lake Louise, Alberta, that area. Haven't gotten any from there yet, but we did them across Western North America and found an estimated time of divergence more than 50,000 years ago, but not much more than that. And so the entire genus, I would guess, is a couple million years old.
Oh, wow.
Yeah. Yeah.
I don't know, I think you had a slide showing that the further north population, which was in blue, being distinct from the stuff at further south.
Yeah.
Could you go back to that one?
Yeah.
I don't know what those numbers are.
Is this the
The
genome-wide stuff?
No.
This?
No, it's further.
it's PGI CO2.
PGI CO2. This one.
Yeah. So I was thinking, okay, so I guess this is all about two kinds of speed of movement. First of all, I don't know anything about these insects, but they apparently have ways of flying around or something. So I'm wondering, well, two things. First thing that came to me was, okay, if this is a more, that's a harsher habitat up there.
Yeah.
Okay, so as this whole thing warms, you're going to expect that the area of the blue is going to start looking more like the other stuff, right? So what you may call the best diversity area now may change because of climate change, and these guys change because of climate change. And the other thing I was wondering about, so that's kind of like this,
Yeah.
we're trying to find these really high hotspots.
Yes.
But the hotspots are going to be modified.
Yes.
And so that's that. And then I was also, these little guys could just go up there. Just fly up there or walk up there or whatever they do. Fly on with birds. I don't know what they do. And then they wouldn't be changing the blue area. Yeah, insects that are in the blue area now would not necessarily be changing, which they probably would. They could also be immigrants coming in with a different makeup, and so that all changes. So I just don't know the speed of these things.
Yeah, well, so I mean, you're asking questions that are spot on, and we were wondering how much overlap will we have, and you know, do you get this part of that? So by the time I finish this, Elizabeth, you know, there's no overlap. So she will talk about 25 years of population data that we have for these three drainages, and
So you kind of know the speed of the change.
we know a couple things. We did one study 1996, believe that? Where we compared the frequencies of PGI in 1996 to what they were in 1988. And that was a period that was relatively cool. The drought had finally ended of the late 80s. And so we had this kind of lot of snow in the early 90s. And the PGI frequencies shifted so that that cool genotype was more common in those Bishop Creek populations. And they're all from, you know, South Lake, you walk up the pipeline and then up towards Green Lake, that's where the transect is. And so in that place where there's the most genetic variability, we see allele frequencies or gene frequencies at PGI shifting north to south depending on the recent climate. So that's one thing. But I think your other question has to do with, well, there were two things. One is the question about, go ahead.
Well, one is if the climate where the blue area is gets
Yeah.
warmer and more tolerable maybe,
Yeah,
then they're going to change, but then that's the first part.
yeah.
And then the second part is, well, some of these guys from lower down end up there because it's now good for them. And so then they mix together.
Yeah, so that may happen. You know, we hardly ever see them fly.
Oh, okay.
But then something would happen, like John would say, I was at Green Lake and there were a bunch there in the fall and then we never saw them again. So we have some evidence that maybe they fly before they overwinter in September or something like that. But we don't see them flying in the summer, and dispersal must be pretty rare. And so we see a lot of retreat and advance of populations. So if we get 3 or 4 more nice wet winters, we'll see them moving down to lower elevations because it's wetter and cooler now and better for them. And then in these dry years, the drought, you know, 60% of our study area, which Jerry established most of the plots in our study area by hiking along and stopping somewhere and getting his GPS out. And we're still going to Jerry's spots ever since the mid-90s. I got off track here. In any case, a lot of these places, we haven't seen any beetles in 5-10 years. So the drought really knocked them back badly. To your other question about, well, the hot spots may move.
With the places with higher diversity.
Yeah, so this is something the CCG people are working on really hard right now, is to use the projections to imagine weather conditions in some areas will persist for the species. And so they're actually modeling that stuff they're busy doing that. Yeah.
It's complicated.
It is complicated, yeah. Yeah,
Nathan, thanks for the great multifaceted talk.
yeah.
I had a question for you about Wolbachia, and I knew that it's present in nematodes and arthropods, but why not more broadly, and they're very divergent taxa, of course, why not more broadly distributed through the animal kingdom, or is it more broadly distributed through the animal kingdom?
So I'm pretty sure that we would know if it were in other organisms, although I'm not sure anybody systematically looked for that. But I don't know. I think it would turn up. Well, any microbiome study that people do, they look for signatures of any bacteria inside anywhere. They would have found it, and they're not finding it. So why? Don't know. But it sure spread quickly when it spread in arthropods. This is not, it might be that 60 million years ago they weren't there. Something has happened to spread throughout the arthropods and the nematodes. And obviously that mechanism I talked about has to be part of the story. So people have been wondering for a while if there are any vertebrate bacteria that could do the same thing. And one of the people who was working on this kind of stuff many years ago, he gave a talk when I was in Switzerland, and he was like, well, there's this one family in Germany where they only produce like 5 generations of only producing daughters. And so he thought maybe that might be a sign that a bacteria that behaves the same way might be in some human populations.
Or just probability.
Yeah, right, exactly. And I know nothing, like that's something I heard 30 years ago, and I have no idea whether anyone's seen anything like it.
That's interesting. But yeah, same with the nematodes, they do a really good job of bumping into everything on the planet, right?
Right.
And so there'd be lots of encounters, in which case they could bridge from one taxon to another.
Yeah.
I forget the biologist years ago said that if you obliterated all their tissue, living tissue on the planet, except for nematodes. You could still see a ghostly outline.
That's right.
Of creatures.
Of everything.
because of their bodies.
Yeah, exactly. Yeah. Yeah.
Can I throw something in here that might or might not be pertinent? But when I got involved in ecology, all the studies were, just 10 years or whatever.
Yeah.
It took to do whatever. Then, of course, we got into climate change, and we started realizing there's been a lot of variation jumping around forever.
Mm-hmm.
So way back when, because I have a geology background too, and so they were talking about these parasitic birds that lay their eggs in other birds' nests.
Yes.
And somebody came up with a number that said, okay, this happened, let's say, 10,000 years ago.
Yeah.
I
Yeah.
was thinking to myself, okay, all you have to do, you don't have to change your body at all. You don't have to do anything. You just have to decide that you're going to lay your eggs here instead of there. That's the adaptation.
Mm-hmm.
So it seemed logical to me that this parasitization has probably taken place many, many, many times. And what we think now is like, oh, here's the first time, finally, in our own lifetime almost.
Yeah.
It happens to be the first. Probably all in this study, which is very complicated, there's probably a lot of stuff that we think is just come about, but I don't know, but actually has come and gone.
Yeah. I think that's definitely possible.
And if it wasn't this subspecies or from variation, it would have been another one,
Yeah.
maybe related.
Yeah.
Yeah.
Well, one thing about those Wolbachia I do know that they're also global. So like we did sequence one of the European Laponica and got very similar Wolbachia as the ones we have here. So the bacteria insect association there predates any of the species that are there now, the specific strains. And my guess is once they're colonized, then they won't get colonized again very easily. And so, when you're talking about those types of birds, in the back there, in addition to the wasps that build the nests that stuff the beetles in there, we've got some what we call cuckoo wasps, which will then sneak in while mom is away getting a new beetle and lay her eggs in the
Which
wasp
is almost
nest.
a freebie. You just have to not get caught by the mom.
Exactly. And they're very active and they run around very fast. Duck in and duck out. And John spent, I don't know if you know John, but he spent like weeks underneath a log watching all these wasps go back and forth and making observations about how many beetles they got and how long it took and stuff like that.
Thank you.
Yeah.
Hey Nathan, thank you very much first off. I have a quick question just about, I know that since you guys have been doing this and I've been part of this working here at White Mountain, that you've seen a decline in beetle population, and I know that big winter that we had, the huge winter, was a big, this with the thaw and whatnot, and just curious, are you expecting to see, a rebound of that? Are you expecting that there is this decline that's still continuing? I'm not sure you're still working on that, but I'm just curious your thoughts.
So in the past, I thought it would be a 1-year time lag. And again, Elizabeth will talk more because we've seen multiple rounds of this. There wasn't a 1-year time lag. They weren't very common in 2023 when we finally got there after all the snow finally melted. And then last summer we thought, okay, we'll see more now. We started to see more in August. When I came back with Cameron in the fall, then we went to some places where they've been rare for a long time and we started to see more. So I think maybe it'll be a 2-year lag, and I hope to find a bunch this summer.
Okay, that's what I was hoping,
Yeah.
I was wondering, hoping as well. Okay, thank you.
Cameron is working on the abundance of the wasp and the fly because they went down with the beetles. And we're going to sort of document the relationship between the abundance of the two, predator prey this year.
Yep. Stay tuned.
Yes.
That's right.
Yeah.
The next 40 years.
That's right. There we go.
So, why was the Permian mass extinction such a big deal?
Yeah, why?
Is anybody familiar with what happened in the Permian extinction?
I don't remember.
The Siberian traps of northern. What do you say?
ultimately the loss of the ozone layer.
Right, so a couple big things. The biggest thing is our 251.9 million years ago, we had about 1 million years of extensive volcanism. A large igneous province erupts in northern Siberia, but not just instead of just erupting, as it goes up, it kind of creates like a cap, almost like our little shield volcanoes that we have around here, where you have like a little salted cap. And then instead of injecting itself up to the Earth's surface, it injects itself into carboniferous units to the sides and essentially spread itself into a bunch of carbon, methane, sulfuric, essentially gas-bearing units, then just cooked our planet, right? So for 1 million years, we just have a nasty, nasty, nasty environment that just kills, 98% of species on this planet. And then things hang on to life in special little windows. So you see everything just go boom. But what I found really interesting was, almost like some of the drops that you see in the late Cretaceous before, then just that right through the middle of all that, the flowering plants are just still just whipping north.
Scooting up.
Everything else seems to be dropping.
Yeah.
They all are just keeping going up. So again, we have stuff to talk about.
Yeah.
Another thing about plants with the peak of that extinction
Yeah.
is that it was at least 5 million years before there were regularly forests. So even plants were down to low shrubs.
Right.
That's all there were, because there were no flowering plants yet.
Yeah. Yeah.
So the plants had no pollinators, so the plant lineages died off as well.
Mm-hmm.
And so
Yeah.
plants basically were just all low shrubs.
And there was this whole sort of radiation of conifer-type plants, gymnosperm plants, that were visited by lacewings.
Yeah.
There's a whole plant pollinator syndrome happening, got wiped out by all that, and so that's why bees and wasps. Yeah, exactly. Yep. Yeah. Okay.
It's like, so you mean to tell me that Wolbachia causes parthenogenesis in aphids, more or less?
Well, see, Owen. So there's a lot of things with parthenogenesis, and with aphids, my guess is yes, but there are other insects with parthenogenesis, especially wasps, things like that. And even that syndrome in the wasp order of insects, where the males are haploid and the females are diploid, and that whole way that they reproduce, because the females produce males without a male. And so that syndrome probably was driven by bacteria. Yeah. Yeah. Walking sticks. There's some walking sticks that, we have to have a special permit because you don't need a male in the terrarium. You just feed them blackberries, and before you know it, there's babies all over the place.
That's really a good thing. I've read about their sex behavior, and it just does not sound very productive.
Yeah.
With, having sex for 3 days straight. So, whoa.
Well, it's better in bed bugs, where what they do is pierce the hemolymph.
Yeah.
Ooh.
So, okay.
I wonder if part of why Wolbachia might be limited to certain taxa of animals has to do with the fact that they're endosymbionts and they're inside the cell with mitochondria. And the mitochondria, there's Wolbachia mitochondrial incompatibility problems, and be something that we are, if we ever get another grant that assess still exists, that's actually something that we expect might be going on, because a lot of, like in our bodies, we don't have a lot of endosymbionts. Most of our symbionts are like inner gut, so then they're not, or they're encapsulated in a vacuole. But the Wolbachia are like, they're in the cytoplasm, they're playing around. So
So are they in vacuoles, or are they just in the cytoplasm?
No,
They're both.
that's not. But they're definitely in other endosymbiotic situations like corals, they're very much encapsulated, right? They're not in contact with the cytoplasm. With the Wolbachia, we're not sure. But the way that insects have their gut is with the Malpighian tubules, and you were talking about how they take up the plant metabolites. That could be another, that's kind of unique to arthropods, how they are taking, how they're taking food in and things like that. So it's possible that in other animals, there's just not the availability. It doesn't mean that other animals don't have symbionts. They may just not have this set of strain of endosymbionts. So.
Yep. Yeah.
Just thought.
Another weird thought that came to me was, okay, why do we have sex? And so it's a very simple way to say it. It would be that, well, we're using up half the individuals that don't reproduce. So why
Right.
do we do that?
Right.
And I always thought, well, it's because all these microorganisms can just reproduce like mad, and so to keep up with their variation, we have to have another individual who we basically just use for that. In your case, you're in between, because you're talking about a male and a female, and now they've got these integrated fast reproducing things, and so they say we don't need the males anymore.
Yeah. Yeah. Exactly.
So it's kind of like another way to think about, you know, that whole question: why do we have or not have sex?
Yeah. I totally agree. I mean, these things are a mix of 3 different genomes, 2 of which are inherited just from mom to her kids, and the other one, the nuclear genome, with half contribution from a male. And so, you know, there's some reasons for example, why mitochondria need to come from only one parent. And, you know, the scenario you described, Red Queen's race is one way to say it, that the host is running as fast as it can because the environment's changing so fast, and the environment that's changing is the environment of the microbes that are inside you, which are evolving so quickly.
So now that we're increasing the speed of change, is there going to be, you know, is this going to make like more of this kind of situation where there's a third thing of genetics? You know,
Who knows?
And
Yeah.
probably it's happened again. It's probably happened over and over because climate has changed a lot,
Yeah.
drastically.
Yes.
Yeah, over time.
Yep. Yeah, I agree.
Anyway.
Yep. All right. So there are bugs over there in the drawer, and I can point them out to you if you want to see them. And other than that,